Hello friends, hope you enjoyed my first previous video and you understood the way of glucose catabolism by the glycolysis. What happens when a single glucose enters through a glycolysis. Now we studied this at the end of glycolysis, the product of glycolysis, end product of the glycolysis is pyruvic acid. Now what is the fate of pyruvic acid? Here pyruvic acid catabolized by different ways depends upon the aerobic and anaerobic as well as fermentative bacteria. Especially here we will go for the catabolism of pyruvic acid in case of aerobic bacteria. Now in this aerobic bacteria which require oxygen for their growth, they use this pyruvic acid catabolism or they perform catabolism of pyruvic acid with the help of TCA cycle. So today in this particular video lecture we will see the TCA cycle, tricarboxylic acid cycle. It is a fueling pathway for getting large amount of energy from this pyruvic acid. Now this TCA cycle is also called as Kreb cycle. TCA stands for tricarboxylic acid cycle. Most of the intermediates of this pathway consist of 3 carboxylic groups. That's why it is called as tricarboxylic acid cycle. Now which are the steps of the TCA cycle? As you know, this is the end product of glycolysis, that is pyruvic acid. Pyruvic acid, and before entering into the TCA cycle, there is a bridge reaction. Pyruvic acid is converted into the acetyl coenzyme A and by the dehydrogenation. There is removal of one hydrogen pair from this pyruvic acid. It was accepted by NAD and NAD is converted to NADH2 and the product form is acetyl coenzyme A with the help of acetyl coenzyme A. Here the enzyme which governs this reaction is pyruvate dehydrogenase. This is the key reaction. Remember, in this key reaction there is production of one NADH2. So this is the key reaction, bridge reaction between glycolysis and Krebs cycle or TCA cycle. Now this acetyl coenzyme A is the intermediate which enters into the TCA cycle. In the first reaction, this oxaloacetate, which is the oxaloacetate which forms in the last reaction of the TCA cycle. This TCA is not pathway, it is a cycle, because in a circular manner there is recycling of the intermediates, or the end product is oxaloacetate is recycled in the same pathway. This oxaloacetate combined with acetyl coenzyme A to form the first intermediate and it is citrate. This reaction was governed by citrate synthetase enzyme. This citrate undergoes dehydration. There is reversible removal of water molecule from citrate to form cis-aconitate. Again in cis-aconitate a water molecule was added and there is formation of isocitrate by the enzyme aconitase. These two reactions are governed by enzyme aconitase. Later on this isocitrate is converted to the oxalosuccinate by removing, or there is removal of these two hydrogen molecules. They are accepted by NAD. NAD is converted to the NADH2 and the intermediate form is oxalosuccinate. Further, this oxalosuccinate undergoes decarboxylation. There is removal of one carbon dioxide from this. This is the position of carbon dioxide which get removed, and this oxalosuccinate is converted to the alpha ketoglutarate. It is the key intermediate. Here this alpha ketoglutarate undergoes dehydrogenation. There is removal of carbon dioxide, not dehydrogenation. Carbon dioxide was removed. At the same time, a hydrogen pair was removed that was accepted as NAD and coenzyme A. It was added by to the product. The product form is succinyl coenzyme A, and formation of one NADH2 is there in this particular reaction. Now this succinyl coenzyme A, it is one of the high energy compound as it consists of coenzyme A attached by a thiol ester linkage. This one is a thiol ester high energy bond. Now this succinyl coenzyme A undergoes phosphorylation. There is removal of energy from coenzyme A, bound by coenzyme A. This was accepted GDP is converted to the GTP and succinyl coenzyme A is converted to the succinate. Then this succinate undergoes dehydrogenation. Again, the dehydrogenation leads to the formation of fumarate and this hydrogen pair was accepted by another reduced energy, that is FAD, flavine adenine dinucleotide, to form the flavine adenine dinucleotide hydrogen dihydrogen. This fumarate undergoes addition of one water molecule by the same enzyme enolase that we have seen in these two steps. That enzyme add this water molecule in at this position to form the malate. This malate further undergoes dehydrogenation, removal of this hydrogen pair. It was accepted as NAD. NAD is converted to NADH2, and finally this malate is converted to oxaloacetate, which recombine to the acetyl coenzyme A. That is how, in a circular manner, the Krebs cycle takes place. That's why it is called as TCA cycle. Now we will see the net gain of ATP. This pathway was particularly called as fueling pathway because large amount of energy was derived from pyruvic acid in the aerobic bacteria or aerobic microorganisms. Now it is a net gain of energy, net gain of ATP through this Krebs cycle or TCA cycle. Now as I told, at the end of glycolysis there are two molecules of pyruvic acid. So when these two molecules of pyruvic acid enters, it gives double ATP. Number of ATPs will be double. So first of all, the reaction number 1, which is a bridge reaction, here we get one NADH2. As I told, from one NADH2, when it passes through the electron transport chain, we get the 3 ATP molecules. So I will write it as a 3 ATP plus 3 ATP in the bridge reaction. Then next reaction where we get the reduced energy is fifth reaction. At the fifth reaction again we get one NADH2. Large amount of reduced energy was generated in the TCA cycle. So at the fifth reaction again there is release of 3 ATP through the oxidative phosphorylation. Then reaction number 7 where we get the 1 NADH2 and obviously it gives 3 ATP. So plus 3 ATP. Then next one is the 8th reaction. In the 8th reaction again there is release of 1 GTP. Here GTP is equivalent to 1 ATP. So the net gain of ATP in that particular reaction is 1 ATP. Then reaction number 9. In the reaction number 9, 1 FADH2 was released. From 1 FADH2, when it passes through the electron transport chain, it will give 2 ATP. So there is gain of 2 ATP in the 9th reaction. Then the reaction number 11. At the reaction number 11, there is release of 1 NADH2. So ultimately it will give the 3 ATP molecule. And now what is the net gain of ATP in the TCA cycle? 3 plus 3 plus 3 plus 3, it is about 15 ATP. So total net gain is the 15 ATP in the TCA cycle. And as you know, there are 2 pyruvic acid obtained from the glycolysis. 15 into 2 is equal to 30 ATP. So 30 ATP is the net gain of ATP while passing this pyruvic acid through the TCA cycle. That's why it is a fueling pathway. Now, which are the modes of ATP generation in that particular pathway? One is this reaction, succinyl coenzyme A to succinate. There is generation of GTP, that is equivalent to ATP. So this particular reaction, succinyl coenzyme A to succinate, is an example of substrate level phosphorylation. And other all reactions, reaction number bridge reaction, then reaction number 5th, that is isocitrate to oxaloacetate. Reaction number 7, conversion of alpha ketoglutarate to succinyl coenzyme A. Reaction number 9, conversion of succinate to fumarate. And reaction number 11, malate to oxaloacetate. And all these reactions, 1, 5, 3, 9 and 11 reaction. In all these reactions, there is production of NADH2 or FADH2, which is the reduced form of energy. And that should be converted into the ATP with the help of oxidative phosphorylation. So this is a way of glucose catabolism in case of aerobic bacteria. Now in next video lecture we will see which are the modes of ATP generation, which are the ways of ATP generation in aerobic, anaerobic and photosynthetic organisms. Thank you.